Greetings everyone, my name is Joe Francis and I'm a professor of biology and I have taught biology for many decades now. I also have degrees in microbiology and public health and in immunology, and I really enjoy studying anything about God's creation and using science to do it and glorifying God in that way. This presentation is about bacteriophages, and bacteriophages as regulators of the human microbiome. This talk is about how, as a scientist who is a creationist, I would approach a new scientific study or idea based on a hypothesis I came up with while doing some research investigation. So this is a talk about how I would approach this research project. Bacteriophages are viruses, and they exclusively infect bacteria. And so that's why they have a special name. They're bacteriophages instead of saying virus. They are viruses, but we call them bacteriophages. And the microbiome is a term that's used a lot today to mean a lot of things, but it basically means all the microbes in a certain environment. So we have a human microbiome. In many cases, people equate that with bacteria, and you can have that usage also. But in this case, we are going to be talking about the human microbiome and concentrating on bacteria because bacteriophages invade microbes or bacteria. Okay? So let's take a closer look at this and see how I came up with this hypothesis. We can also talk about the microbes on Earth, back up a little bit, and talk about a bigger concept here. And in an area that I've been working on, I propose that microbes on Earth are everywhere. Okay, there's so many that they form a matrix. There's hardly any microbes that can be alone in this matrix because they divide every few minutes, and so you can actually make your own community. But we find the biomatrix on trees. We find the biomatrix deep in the ground. We find this biomatrix of microbes in the ocean, and so on. And from a creation perspective, the question would be, God designed and made everything, so what is this biomatrix doing? What is this interlocking matrix of microbes doing on Earth? By and large, they're doing some good things, even on a global scale. But as we talk about this, we can continue to talk about the terms here. So microbiome is all the microbes that can live in a certain environment, often referred to as concentrating on the bacteria that live there. A virome is all the viruses that live in a certain area or place. So we can have a human virome. There are viruses that live on the human body. We can also say the virome is a part of a microbiome because microbiomes are made of different kinds of microbes. So that's how we use those words. All of them together, we try to fit into this biomatrix concept that they are forming a matrix, a life-supporting matrix on Earth. We can talk about numbers, and without really understanding, without doing some work here to figure out what that number means, that doesn't look so impressive. But 10 to the 29 is a huge number. That's billions times billions times billions and onward. There are a lot of microbes in the biosphere on Earth, or this biomatrix. You notice the virome, there are actually more. And that kind of makes sense because most of the microbes of the bacteria and algae and fungus and all those things are populated by viruses. So we could forecast that there's more viruses than bacteria and other microbes on Earth. But what are all these viruses doing? They're invasive. They tend to destroy things. They can cause disease. Well, if we look at it from a creation biology type view, that God probably had a good intent for them, we find that they're mostly delivering things. They can deliver things to bacteria, they can deliver things to the human body, they can deliver things to other creatures, to plants, and so on. So why do we have these delivery agents, and what are they doing? So we're going to concentrate on the human virome, the viruses that live on us, and I'm going to make a proposal that these bacteriophages, play an important role in protecting the human microbiome. Okay, but let's look at the human virome first, the viruses that live on the human body. What's interesting is that we can look at three groups. We can look at the eukaryotic virome, and the eukaryotic virome are viruses that live on our typical body cells. Our typical body cells are called eukaryotic cells, okay? And then the other two categories are basically bacteria categories, but we have special names for these bacteria groups. Eubacterial bacteria are the common bacteria that we know about, like E. coli and Staphylococcus that cause staph infections and things like that. Then we have the archaeal virome, and these are extremophile bacteria. They tend to live in extreme places. Not too many in the human body, but we do find some in the human body where there's not a lot of oxygen, and that's an extreme place. So when we look at this, what's interesting is the eukaryotic virome. The viruses that live on most of our cells, our red blood cells, our skin cells, our brain cells, they're transient. They don't stick around long. Our immune system takes care of them, or they destroy the cell they're in. And then if they have no place to go, they're not going to survive. They're very transient. So we also call these viruses in general animal viruses. And this includes humans. And in biology, that's often the case. Humans are considered animals. So these eukaryotic viruses, or animal viruses, are the cold virus, the flu virus, and so on. They're transient because our immune system clears them out. Curiously, there's like two viruses. One's called a JC virus that might have a human origin. In other words, we can't find an animal that presented it to us, because a lot of flu viruses come from birds and a lot of other. So this is really interesting. It's an interesting point by itself. We just don't have a lot of animal viruses or anything we can call human viruses, except for a few. So when we look closely at this, we also see that we don't have a lot of archaea viruses that live in the extreme bacteria, but we have a lot of viruses that live on our common bacteria microbiome, okay? And so the basic counting we've done has changed over the years. How many bacteria live on us? Probably at least one for every one of our body cells. And the basic, general-sized human has about 35 trillion bacteria, or cells. And one bacteria for at least each one of those cells. So we have 35 trillion bacteria growing and living on us. And if each of those bacteria have a bacteriophage inside of it, they tend to invade and incorporate themselves, then we have 35 trillion bacteriophages, but probably more, because they're small and they can fit inside, many can fit inside the cell. So this is kind of interesting that the animal viruses, the flu viruses and so on, don't stay around. Now, there's some like the herpes virus that can stay in the human body for a long time, but again, most of them are transient. That means the main virus on us are bacteriophages. And in a sense, we are more bacteriophage than human because they outnumber our cells. Quite interesting. So I was just teaching microbiology and thinking about this. Why bacteriophages? What are they doing? What are they doing for humanity or the human body? What is their design feature? Again, this is just a chart showing the numbers of animal, plant, and archaeal viruses in the human body. There's still a lot of them, but not as many as the bacteriophages. We didn't talk about plant viruses. Actually, one of the viruses that dominates your gut are plant viruses. And we can only come up with an idea or an answer to that because we eat a lot of plant material. But again, they're transient. They're transient. So let's look at phages a little closer. Let's look at their structure. They can be very different from traditional viruses. Most animal viruses have a lipid coat. They tend to be roundish. But phages take on many different structures, in part because a lot of them don't have a membrane on the outside. They have a protein coat. And proteins are stiffer than lipids. Okay, they're stiff and they can cause these shapes to form, like icosahedral shapes here. Kind of look like little spaceships in a way. Even have landing gear to land on cells and so on. We do have some lipid enveloped ones, okay, but a lot of rigid shapes due to the protein surface. We do have some that are filaments, though, We have some filaments that are curved at the end and indicate a certain bacteria, like the Ebola virus. It's called a shepherd's crook. It's intriguing, the different kinds of shapes you have here for these creatures that invade bacteria. Why those shapes? We don't know, but it's fascinating. So here are some facts about phages, and some of these come from a professor in California who has studied phages almost exclusively, especially those that populate the oceans. And he has come up with a calculation about how many phage infections there are globally. I have no idea how he did this. But he is saying that there are 10 to the 23rd infections per second of phages going into bacteria. Wow. 10 to the 23rd infections occurring every second globally. So phages are out there. They're abundant. Again, what is their role? What was God's purpose for them? They're involved in the global carbon cycle. They actually help. One of the most abundant bacteria on Earth is a photosynthetic bacteria. And photosynthetic bacteria take in CO2, give off oxygen, so they're part of a carbon and oxygen cycle. And these viruses actually help these bacteria survive in the ocean. These phages help them survive. And so they play a part in how much carbon is being uptake into living things and how oxygen is produced on the Earth. This Dr. Rohrer is his name, also has looked at the phages in the ocean and looked at their internal contents, their genetics, and he finds that they're a repository of genes. So you can scoop up some ocean water, isolate some phages, and you can find genes for making vitamins. You can find genes for stress response proteins. You can find genes for DNA, or what we call nucleic acid metabolism, how DNA is put together. We can find genes of virulence factors. Virulence factors are factors that bacteria use to cause disease. We can find chemotaxis proteins. This gets really weird. Chemotaxis is how bacteria actually move toward a food source. They tax toward the chemical given off by the food source. It almost seems random that we can scoop up ocean water and find proteins that may be found in humans and found in bacteria in these phages. He kind of gives the story, as you read his work, that there's like a bank or a repository of genes in the ocean in phages. We don't quite understand it. We just know it tells us they get around a lot. They get around a lot and pick up these genes from the bacteria they invade. He also says, here's the name of the professor, Dr. Forrest Rohrer at San Diego State University, who's done some intriguing work. He says there are 2.5 billion novel phage-encoded genes in the biosphere. He just says it's like this big filing system. And you think about it, here's God's design. Is there a purpose to this? Is it like a backup mechanism? The problem is we can't get those genes from phages because they don't invade us, but we could get them from the bacteria that invade us because they invade the bacteria in us. So it's just intriguing. It's just intriguing how numerous they are and how they just kind of pick up genes and carry them around. Another interesting thing about phages is they have practical uses. So if we look over here at pest control in this diagram, there's a lot of insects that can destroy plants, or they cause them to not look so good and wilt and not be of any use in agriculture. But if you look closely here, if you treat them with a phage, okay, or a bacteria, what you have is, a phage. What happens is the insects get smaller. In both cases, they're showing the insects getting smaller here. And what we know is this: a lot of insects work with bacterial partners. A lot of insects work with bacterial partners, and they're healthier, and the ones that invade things become more invasive with these bacterial partners. But remember, phages can destroy bacteria. They can actually They actually invade the bacterial cell, make copies of themselves, and break out. So what they're saying here is, if we have insect pests that use bacteria to make them more invasive, if we treat them with phages, the insects will actually get smaller and less invasive, and we'll have healthier plants. Same thing in the food industry. You can have a lot of bacteria that might cause human disease on our food. We could spray the food. A lot of these are not being done yet, but they're forecasted to be done. We could spray the food with phages that break open those bacteria and kill them. Down here there's a lot of information, but I'm going to go skip to the very bottom here. It says we can treat dysbiosis. Dysbiosis is a term that means your good bacteria, your good microbiome in the human body is not working right. Something's misfunctioning. And it's probably because there's a bacteria that's not supposed to be there. So we could use phages to get rid of that bacteria that's not supposed to be there and cure or reverse dysbiosis. We even attribute cattle on Earth to global warming. I'm sure it's debatable, but there is a premise there, and that is that cows put a lot of carbon in the air through production of methane. Their gut is basically a huge fermentation vat, okay, and the end result is methane. So we could decrease methane in the air by giving cows phages to kill some of those bacteria. I don't think you want to kill all of them, or the cow couldn't digest its food. These are projected ideas for how phages could be used in the future, very practical. And since they don't really harm us, there's some possibly good solutions here. So what are all these phages doing in mammals, in humans? The phages that live in our bacteria, what are they doing? What is God's purpose here? We could think about them being involved in population control and host protection. So here we have the lining of your stomach or your intestine. And we know, because if you see projections coming off a cell, in your gut, those projections offer more surface area to take up nutrients from what you have eaten. So this is the inside of the gut right here. We have a kind of a green-yellow mucus layer, which is actually good. Prevents bacteria from getting too close. But our microbiome kind of lives here. Lots of bacteria live right here that are not shown. What we do have is an invader, a bacteria that's pathogenic, which means to cause disease. And phages could very well be attacking those bacteria and destroying them. Okay, so they could help the population control of the microbiome or control bad bacteria that live with the good bacteria. So that would give us what's called host protection. We are the host of these organisms and we can be protected. So yeah, phages could help control bad bacteria or the overgrowth of bacteria in our body. So I've been thinking about this for many years, and a few years ago I presented this idea at the International Conference on Creationism of something called the Phage Cloaking Model. I started thinking about the idea that maybe one of the ways that our good bacteria are protected from our immune system is through phages coating the exterior of the bacteria. So our immune system works best by looking at the surface of an invader and saying, is that foreign? Is that foreign? They take a receptor out. You can kind of think of it as tasting. The receptor is kind of tasting, that's a foreign molecule. Let's bring some white blood cells over. Let's destroy this invader. Well, wait a minute. Some of our good bacteria have similar molecules on the surface, and they're not making us sick. And we're finding out today in medicine, you don't want to lose all your good bacteria. So I started thinking, maybe phages are coating our bacteria, our good bacteria, helping them to persist in the human body. The Phage Cloaking Model flows from the biomatrix creation model. Again, the idea that there's a matrix on Earth, okay? This is part of the matrix. And there are two parts to the proposal that I want to show you. And two parts are there's a microbiome genome gardening. So the microbiome are the good creatures, the good bacteria that live on our gut and on our skin. and they have a genome. A genome is basically their DNA. Okay, the genome is all their DNA. And I'm going to make a case that phages guard the DNA in our good bacteria in the microbiome. And I'll show you how in a minute. Also, they can shield. They're very good at coating bacteria. So there's a second part to the phage cloaking model, and that is they could shield the surface of the good bacteria and our immune system, making our immune system blind to it. So let's look at what a phage actually does. We're going to look at what a phage actually does to a bacterium and then go back to the cloaking model and see if it makes sense. So if we start right here, we see a green surface on a rod-shaped bacteria. All bacteria have a circular piece of DNA. In humans, we have strands of DNA, but they're circular. Here is a phage injecting the red part, which is its DNA or RNA. And it does sit on a cell like a spaceship with those fibers. It does move that middle part down like an injection port. And it injects its DNA. And it incorporates into a bacterium. And it actually can live that way. It can actually go two directions. So we're going to see how it goes on this top cycle called the Lytic cycle first. So it incorporates into the DNA. It can stay there a few hours, a few days. And then that piece of DNA can come out, that red piece can come out and start making copies of itself, okay? It has DNA that makes its own proteins to make its copies or borrow the machinery of the bacterial cell. So now you see copies of red DNA, and then those DNA, and then it starts making copies of its coat protein. So if everything in blue is a protein on the surface of this phage. So the DNA goes in and it has the message to make more of itself and the proteins that coat it, the outer coating. It actually makes proteins to assemble the little phage. Pretty amazing, really. It's like this thing has a brain. And then it puts all these pieces together to make new phage. And I always thought by sheer number, they exploded out of the cell. That's not true. What we now know is they make a little protein that actually pokes through the outer surface. And that's called lysis. Lysis means to break open. So that's the lytic cycle. What's it going to do to the host bacteria? It's going to destroy it. It's going to kill it. What is the advantage here? Good question. Maybe it picks up some DNA that it could help with its survival. But there's another life cycle from the same virus. The same virus can invade the cell, put its DNA inside, but not break it open, not come out of the DNA. It just actually stays there and gets copied into new circular DNA and is transferred on. That's called lysogenic. Or we could say it's non-lytic. It's not lysing or breaking open. This is interesting. So I started thinking about this. And I went back and looked at the husband and wife couple who really discovered how these cycles worked. But long before they had discovered how these phage cycles worked, they were studying the typical E. coli bacteria, which has been the most studied bacteria in microbiology. And they were studying the E. coli growing on plates. So E. coli grows on agar plates. Agar is a polysaccharide from seaweed. It just happens to be really good for growing bacteria. They were growing bacteria on plates, and it didn't look like this plate. The plates they were using had agar, and when they grew bacteria, they found white spots, because every white spot was a colony of bacteria. One bacteria starts growing on the agar, divides, and divides again, and forms a circular colony. But what they noticed over time is sometimes they would get colonies all over the plate, but sometimes there would be a piece of the plate where the colonies could not grow. They're all gone, like something was preventing them or breaking them open. And so they said, let's study this a little further. Their whole goal was not really to study viruses. Their whole goal in the 1940s and 50s was to figure out the genes inside the E. coli. What are the genes? They're just defining what a gene was then too. What's a gene and where are they? So they were trying to find tricks for looking at the DNA. And they just kind of I think it's more by accident, they discovered that phages were blowing up some of the bacteria. So then they made lawns of bacteria. Instead of waiting for them to grow to make these colony spots, they took lots of bacteria that were already grown. They then kind of painted it over the agar. And so there's a lawn of bacteria. So wherever you see light green, that's growing bacteria. Wherever you see a circle, there's no bacteria. And what they figured out is that's evidence of a phage destroying those bacteria and creating what's called a plaque. Now, that wouldn't be just one destruction of one bacteria. They're destroying millions of bacteria as they spread through that, as they start in the middle and break open some bacteria and they spread out, and then they seem to kind of stop, depending on when you stop the experiment. So this is really interesting. They started seeing these plaques. So here's the phage they used. It's a very famous phage called the lambda phage, the Greek letter there. And here's the lambda phage that they were studying. And so now, based on their work, I had an idea from their work, and now this is many years later, 80 years later, I start looking at this through their eyes a little bit, even though they were looking for something else. And I started thinking, the good bacteria, so imagine this is E. coli in our gut. There's a lot of good E. coli in our gut. What do? They actually make vitamin B12 for us. They can make vitamin B12 and help with digestion and things like that. Okay? E. coli is not the most dominant gut bacteria, but it's one we know a lot about. And I'm thinking, okay, these E. coli have a phage in each of their DNA. And it turns out that if you go back and look at this model, let's back up again here. I was thinking the only way that these phages can make a plaque, okay, is if those phages have integrated into the bacterial DNA. Because remember, before these plaques, okay, form, everything's smooth, and then maybe a day later they see a plaque. And in fact, I forgot to tell you what Esther 𝘕𝘕berg did, is she used a UV light and she got more plaques. That's really interesting. She with her husband, is figuring out the model here that you can stick a piece of viral DNA in the bacterial DNA. But if you shine UV light on it and mutate that DNA, the virus pops out. That's really interesting. It's as if the virus is saying, this is bad, I want to destroy it. So I picked up on that idea, and I developed a model that in the human gut, we want to keep all our good E. coli, okay? And let's say and lambda phage is everywhere, lambda phage, I should say, is everywhere. So imagine all these E. coli's have the little red DNA from lambda phage in them. And it's kind of like an alarm system ready to go. So any carcinogen like UV light or any chemical that causes mutation could cause that little lambda phage to come out and destroy those bacteria. And I'm thinking, this is really cool. This is not what the 𝘕𝘕bergs were trying to discover. They were just trying to figure out what was in the DNA of the bacteria, and these viruses were doing something interesting to the DNA of the bacteria. I started thinking in a different way, because in a creation model, I know there's good created bacteria living in our gut, but what if they got mutated? And what if they started dividing out of control and they outgrew all the good bacteria or destroyed the good bacteria? And what if they made a product that was toxic because their DNA is now mutated? I thought, this is really, really cool. This is really cool. Okay, so the carcinogen, again, imagine the assumption here is the E. coli in the human gut has the lambda phage integrated. Here's the lambda phage, remember, and here is its integration, the red. I'm assuming, because lambda phage is so good at integrating, it's there. A carcinogen comes around, and the bacteria that get mutated are lysed by the bacterial life cycle. going into a lytic phase. So this is really neat. Now, I don't have any direct evidence. So this is why I'm in a hypothesis stage, but I hope in our creation and science world, we could have these hypotheses and some other creationists might say, hey, I'm working on this model. I can do this experiment. But this is really cool. So one of the things that surprised me, though, is it's surprising that nothing is documented about lambda phage protection of the microbiome. No one has looked at this. Why? Because maybe not thinking like a creationist is thinking about good bacteria. Surprising that nothing is documented about lambda phage protection of the microbiome, especially since proteins which are known as guardians of the genome exist. So we actually have in our bodies a protein called p53. And whenever it is disturbed, it tries to destroy a cell. We already have a guardian in our cells, but this guardian's not in bacterial cells. So what if the lambda phage is like this guardian p53? So that's the genome guarding. Remember, genome is a DNA, and the genome guarding is that the lambda phage, once it sees something mutating in the cell, comes out, forms the viruses, and kills the cell. This is the immune shielding part of the hypothesis. And I came up with this idea because I'm looking at my virology text, okay? A text about viruses from my undergraduate days. And I went back and looked at the pictures, and just about every picture, you have these phages. These are electron microscope images because things are so small here. You find, you know, maybe 20 or 30 pictures in my book of phages in the process of infecting a bacteria. The surface, you can imagine the surface here is kind of under these phages, and just they look relentless in their ability to coat the bacteria. But here's what I couldn't figure out. A lytic virus should go in and destroy a cell probably within seconds, maybe minutes. So how can I see this? How can I see all the viruses that are left there? Why didn't the first phage go in and destroy this thing? And so I read more about phages, and I found out that phages that coat a bacteria, they're looking for an entry point. They can actually undergo something called walking, and it's just from, they're so small, it's just from the activity of the fluids around that their fibers can actually come off and go down and come off, and they can find, and then they eventually find an entry port. That's even more interesting. But what got me is, even if this cell gets killed, we find it's coated for a long time, okay? Or maybe this bacterial cell doesn't get destroyed. So I started thinking, wow, what if this is some kind of coating activity going on? Let me show you more about the coating because it's fascinating. So here is another E. coli-like bacterium. Here are all these what are called fimbriae are pointing out. Some are called pili, some are called rotary motors or flagellum. So bacteria have at least 3 appendages they can stick out. The fimbriae are actually very stiff, and they can be used for locomotion. They actually bend like an oar. They twitch. It's called twitching motion. If we found a flagellum here, it's a rotary motor. It spins that filament and can actually push the cell in different directions. And then the pilus is often a hollow tube. It's a third structure, and actually it can actually send DNA from one bacteria to another. You kind of get the impression here that viruses and bacteria like to move DNA around or capture DNA. Really interesting. So I don't have the images that I want to tell you about, but I drew one here. So they're incredible microscopic images. Some are hard to get a hold of because they're protected by copyright. So I found an E. coli picture, and here's one of these fimbriae or pilus or flagellum, and sure enough, it's coated with phages, just absolutely coated. You can't see the rod. Here, these are, I think, uncovered. You can't see any uncovering. And at the very end, there were two filamentous phages blocking the end. I'm going, what in the world? So I'm showing you an appendage that's uncoated, and I'm showing you a surface which is uncoated, but what I found in the picture is everything was coated. Everything was coated, and the cell was not broke open. What is going on? So this is part of that immune shielding idea, or the cloaking idea. So again, you have phages guarding the genome. If there's a disturbance, oh boy, kill this cell. We have phages coating the outsides. This is what I got excited about, and I wish I had money in a laboratory to investigate more. But let me show you more why this shielding is important. Here is really why this is important. So let me explain some things here. This is a rod-shaped E. coli bacterium here, and we're looking at a cutout section there, and we're exploding this image here of that cutout section. And what we find are two membranes. Okay? There's two major classifications of all bacteria. Those that have two membranes and a cell wall in the middle, and those that just have one membrane and a thick cell wall. And there was a professor by the name of Dr. Graham back in the 1800s. And he came up with a Graham stain. And he found out that when you put the Graham stain into these bacteria that have a double wall, like E. coli, or double membrane, and a little wall, the stain came out. He called it a negative stain. And they applied his name to it. So it's a Gram negative stain. If you find a bacterium that just has one membrane and thick cell wall, once the stain gets in, you add something to kind of make the stain a little bigger in both these cases, and the stain took. So the stain stayed inside the cell where you had the thicker cell wall, only one membrane, and that's called a gram-positive bacterium. And this is actually helpful in medicine today, because a doctor might say to another physician, this patient has a gram-negative infection. And that can imply something, because gram-negatives can be really bad or have a classification of their infection that might imply skin or tissue or something. So we still use Dr. Graham's staining today for very good benefit. So here's a gram-negative cell, and this could be an E. coli, which is, yes, gram-negative. And you have these two bilayers with the yellow and the tails coming up. Those are membranes. But what I want you to notice are the proteins stuck in the membranes. And then you have something called LPS, lipopolysaccharide, which is a phospholipid with some carbohydrates on it. Okay, so here's the important point. If we take any one of these proteins or these carbohydrate chains and we inject it into the blood of an experimental animal, or say a human, you can cause death. It might take some hours and even days, but it's horrible. It turns on the immune system to such a high degree that your immune system basically starts shutting down the body, saying there's something terrible here. I got to lower the heart rate. I've got to do this. And in fact, this is when this happens from a gram-negative cell, and it can happen in the human body in patients, it's deadly. And what happens is the body says, there's a really bad bacteria in the blood. It used to be called blood poisoning. And over time, what happens as you try to treat the patient, your physiology is saying, conserve the brain, conserve the organs. The blood pressure drops low, and fingers and arms and legs and toes turn black. And in order to save some of these patients, you have to amputate. This is called gram-negative sepsis. I'm going to come back to this. Let's just go forward here. So gram-negative sepsis is this medical condition in which the entire body experiences inflammation as it tries to fight off an infection that has made its way into the bloodstream. And LPS, this guy right here, is one of the main triggers of that. So here's what's really fascinating. All your good E. coli have LPS. Why aren't we dying? why aren't appendages turning black, and so on. And let me just say too, there's over 100,000 deaths each year in the United States from gram-negative sepsis. There's actually gram-positive sepsis too. So this is interesting. Our good bacteria retain this structure, but what protects it? How are we protected from it? Well, I think in the gut, it's a mucus layer. It's a pretty amazing layer. And an immune system that basically says, you're okay there. There are immune cells that reach up across that mucus layer. But I'm adding a third idea, and that is the phage cloaking idea. Okay? Could it be that our good bacteria are so coated with phage that our immune system can't see it? Now, still, if you took LPS with phage and injected, I'm doing an experiment in my head right now. If you took LPS with phage and injected it, it still might be toxic, but maybe it wouldn't be. So this is a new idea, so I'm right now kind of entertaining some more hypotheses. I have no idea. I have no idea if the phages actually protect us from gram-negative sepsis. Because I think there's other barriers that have to be breached. But what if it did? What if it did? Again, this is a horrible, horrible illness. Thankfully, doctors today, if they get to it fast, they can try to save the life. But amputation is a reality. So let's go back to the proposed model. I'm going to add a little more here. So imagine the white. So here's the E. coli, the blue E. coli. Now imagine the white squiggly lines there. Those are the phage coating everything. Okay? They're coating out on that outer membrane of the E. coli. They're coating the LPS, that lipopolysaccharide. That's very terrible if it gets loose. And so they're coating the appendages that come off the bacterium. Okay, it's just an idea. And helping, but these old E. coli are still functional and doing good things, we think. That's another hypothesis. Is the coating causing them not to make their vitamin B12? A carcinogen comes in, a mutagen, or just normal cell death. Okay, and I told you, you're going to have lysis in these two cases, because remember, inside there's a DNA from the same phage, and it's going to break open these cells. But also you have cells dying, and they could fall apart, which I believe they do, and they could get into the bloodstream. So now they have this coating around them that tells our immune system there's no LPS, there's nothing here, don't get alarmed, we're just cleaning up. And then these will probably eventually break open in the same coating here also. So that's it. That's the phage cloaking model. It could be totally wrong, but it makes sense to me. So what we have to do in science, though, we can have a nice idea, but then we have to test it. And I would love to see this tested in an animal model, of course, not in a human model, because that if we do release some of these things in our experiments, let's go back for a minute here. If we do our experiments and release these, it's going to be deadly. So this is a model that flows from a creation idea that there are good bacteria and there are good phages. And perhaps they work together to maintain the microbiome by protecting it from our own immune system. Let's look at some conclusions. Phages are abundant in mammals and coexist with the microbiome and appear to maintain microbiome populations. So we do know that phages are on all our good bacteria, and they're not completely wiping them out. In fact, they could be helping to maintain them. Phages possess designs to thoroughly coat disease-causing molecules found on the surface of commensal bacteria. Commensal is another way to say good bacteria. Okay, phages possess designs to thoroughly coat. Phages are found primarily in a lysogenic, not lytic state in the mammalian microbiome, preserving the microbiome by guarding the genome. Now, because they integrate into the genome. Now, how can I say this? I can say this for sure. Phages are found primarily in a lysogenic state. How can I say that for sure? Because if it was all lytic, you wouldn't have any good bacteria left. So we can pretty much say that for sure. Phages are found primarily in a lysogenic, meaning they're incorporated into the DNA, right? They're not making hundreds of copies and bursting out. So this is interesting. This is interesting too. This is what we can say. There's a fact here, and that's pretty cool. So, but could they be guarding? That's what we don't know. Phages help to protect and preserve both of their hosts, because if you preserve the microbiome, you're helping to preserve the mammalian body. Okay? So phages help protect and preserve both the mammalian body and the microbiome. Phages possess designs to maintain commensal microbiome bacteria, but also destroy pathogenic invaders. And we would have to look at how they discriminate. I don't think that's known yet. Based on observations of E. coli, multiple species of phage can attach to a single type of bacteria, promoting the thorough coating of the microbiome bacteria. Yes, I didn't bring this up, but in those pictures, what we have, and looked at more photographs of electron microscopes, and this is well known, you can have multiple kinds of phage of different heights. This kind of blew me away. You have a tall phage, a middle-sized phage, and a lower phage, as if that coating was meant to be very, very thorough. Okay, if something got past that first taller phages, you would have a secondary level. So it gets even more intriguing here. We propose that once lysed, microbiome bacteria parts and virulence factors can remain coated by the phage, preventing life-threatening sepsis reactions. So remember, sepsis is your immune system going haywire. Could they be coating them, our good bacteria? Okay, so epilogue. An epilogue. And this is interesting. What we have here is a young lady by the name of Victoria Gray, who had a life-saving treatment for sickle cell anemia. And why am I bringing this up? Why I'm bringing this up is because we are using Esther Lederberg's finding from 60, 70 years ago, and now using that in a way to perform medical treatment. Now, what's going on? So let's go over to the phage. Here's a phage injecting its DNA. What we also know is that sometimes phages don't fully incorporate into the genome. They don't fully infect, because bacteria have another trick up their sleeve. Bacteria have this blue enzyme called Cas1, Cas2, which, if it gets activated early enough, it can stop this DNA from incorporating all the DNA into the bacterial DNA. This is really intriguing. What it does is it cuts pieces out. It puts them in the DNA, and that acts as a memory for the bacteria. So if it gets invaded by this green DNA phage, okay, it has a memory here. And the same little cutting tool, okay, will pick, it says you're being invaded again. Let's pick up the green one and go over and match. Oh yes, you're being invaded by this phage that has this green. Destroy it. So this gives even more intriguing. Esther Lederberg finally showed that phages can incorporate their entire DNA into the bacterial DNA. But now we know, with a little more investigation, that we actually have what's called a gene editing system. So this thing can cut at various places in the phage DNA, and it can cut in various places in the bacterial chromosome. And guess what? Now we're using this same enzyme to cut into human DNA and do gene editing to save lives. So here's the story. So individuals who have sickle cell anemia have red blood cells that just don't work properly. And they don't work properly because they have a single mutation in the gene that makes hemoglobin. So if you remember what hemoglobin is from your biology class, hemoglobin is a carrier of oxygen in the blood. It's very important. That's why we have a lot of blood cells, so they can take oxygen to wherever it's needed in the body. Oxygen is used by all cells to make energy. So in sickle cell anemia, you have one mutation, and it mutates the structure of that hemoglobin, and it kind of twists and whatever, and it gets sticky to the membrane of the red blood cell. The red blood cell collapses into a sickle shape. So that's where we get sickle cell anemia. It's horrible because it's a disease you basically live with, but you're in pain and you only have so many hours a day where you can do things. And this Victoria, or Mrs. Gray, she had children. Her and her husband had children, and she only had a few hours a day of stamina, of energy to take care of them. So they came up with an idea a few years ago that we could use gene editing to cure genetic diseases. And this is what they did with hemoglobin. So in Mrs. Gray, they took her bone marrow out, which produces red blood cells. And they saved some and they destroyed the rest because they didn't want any more bone marrow making sickle cell red blood cells. And they took that bone marrow, and it turns out when we're babies, just before we're born, we have something called fetal hemoglobin. So there's fetal hemoglobin and there's adult. And guess what? This is really cool. Fetal hemoglobin, it doesn't suffer from the same mutation. It's whole. It's whole. It doesn't suffer. So what they did, and this is very scary to be one of the first patients to do this, they take all that bone marrow out, and they don't do it that way. They take some of the bone marrow out to keep for a while, and then they irradiate the rest of the bone marrow, okay, and then try to remove some of that. So you can't make the sickle-celled red blood cells anymore. During this time, Mrs. Gray would then have people giving her blood to be transfused, while what they would do then with the bone marrow blood they saved, they took this system, they took the Cas1, Cas2 enzyme, and they told it to go over to the stem cells in her bone marrow that make red blood cells, and they edited. There's actually, so what happens in your stem cells as you age, there's a protein that shuts down the fetal hemoglobin. So what happens is they found out what protein that was, and they genetically edited it. They just used the scissors this time. See the scissors right here? They just used the cutting ability to Cas1 and said, go over and cut that protein that's turning off fetal hemoglobin, and her bone marrow cells started making fetal hemoglobin and not adult hemoglobin. Okay, they stopped the adult hemoglobin, and she started making, for the first time, red blood cells that were perfectly shaped with her own fetal hemoglobin. And it's an incredible story, and it's so fascinating that we are using what phages and bacteria have taught us, right? What they have shown us is that there are enzymes, there are editing enzymes in the bacteria that can alter. So this little phage and this little system that we knew about for 60 years, that it can cut DNA and put it into the bacterial DNA, now we can use it for gene editing. She survived. She is doing very well. This is, I think it was done in 2023. Here is her testimony. The life that I once felt like I was only existing in, I am now thriving in. Gray, who is deeply religious, told the audience of scientists, doctors, ethicists, and others at this conference, bringing many to their tears. I stand here before you today as proof that miracles still happen and that God and science can coexist. Victoria Gray, cured by gene therapy of sickle cell anemia, speaking at the 3rd International Summit on Human Genome Editing. It's fascinating to think that God made these little creatures to teach us. They're instructing us, and we can use that knowledge to cure drastic diseases. And we're just on the cusp now of using gene editing in humans. Thank you for listening.